I'm Ihsan. I'm a first-year PhD student at the University of Cambridge. And I'm here today to talk to you about cell division and how things can go wrong. When you think about it, the human body, your body, is an amazing piece of machinery. Within it, 50 trillion cells are tightly controlled so that everything works together in perfect harmony. When you think about how these 50 trillion cells all came from one single cell, the result of millions upon millions of cell divisions to form a living, breathing human being, it's just, I can't describe it, it's just amazing. But the bad thing about the situation is that if one thing were to go wrong, if one cell was to go rogue and not divide as properly and precisely as it should, the results could be catastrophic. The results could be cancer. Now, personally, I'm fascinated by cancer. I like finding out how things can go wrong and result in a tumor. But it's not just me. Cancer has been a big deal recently. Together with obesity and its associated diseases, such as diabetes, cancer is one of the major causes of mortality in Brunei. In the past decade, there has been huge efforts worldwide to study cancer, including, as Professor Tong mentioned earlier, UBD. In Cambridge, I am based at the Department of Genetics. Research interests within the department are varied and include microbial and population genetics, systems biology, developmental biology, and cell biology, which my lab is a part of. The lab is actually funded by Cancer Research UK to study how cell division is carried out properly in human cells. At least 100 different components are known to be involved in cell division, but at this point in time, we're still not sure how they all fit together. So in today's talk, I will talk to you about a family of 4 of these components. Many of you may not realize it, but cell division is an integral part of your body's day-to-day functions. Every day, probably hundreds of cells, which are falling apart, are recycled and replaced by stem cell division. When you get injured, cells in your skin divide to replenish the damaged areas, and when you have an infection, immune cells within your blood again divide to help you fight off that infection. So before I go into my proper talk, I'd like to give a brief background on cell division for the non-biologists present here today. Basically, cell division is when one parent cell divides into 2 daughter cells, which are genetically identical to each other. Before division, the DNA in the form of 23 chromosomes in humans must be duplicated. And during division, this DNA must be equally separated into 2 equal sets. Failure to separate properly will result in unequal segregation of chromosomes, and this is something that we see a lot in aggressive tumors. I'd like to show you a movie of this. This is a video I took of an actual human cell, magnified using a really fancy microscope. Here you can see one parent cell dividing into 2 daughter cells. In this movie, green is DNA, and red are these rope-like structures called microtubules that attach to the chromosomes and separate them. How is this force of separation generated? We know nothing about it. How do the rope-like structures form? Again, we know nothing. How do the microtubules find the chromosomes and attach to them? This is something we know very little about. And so, my goal is to study a family of components called MARC, which are thought to play a role in regulating the behavior of these red structures. As your body goes through its everyday, day-to-day life, the cells in it require small molecules that we call proteins to both carry out its function and to control it. MARC is a family of similar proteins. And it's actually quite interesting to study because it's been implicated in many diseases, as you can see, such as Alzheimer's, autism, and cancer resulting from bacterial infection. Animal studies have also shown it to cause insulin hypersensitivity and immune cell dysfunction. But its role in cell division in cancer is not known. As geneticists, what we like to do when we want to find out what something does is to get rid of it and see what happens. And so, to study MARC, I remove it from cells and look at their fate in cell division using a microscope. To do this, we use a technique called RNA interference, or RNAi, which will remove specific components from a cell. It's a very clever technique, favorite of geneticists, and the people who developed it actually won a Nobel Prize a couple of years ago. Now, you can see here in that top row that while normal, unperturbed cells still have the MARC signal, cells which have had MARC removed in this bottom row here have lost the signal. We take this to mean that, well, the cell has no MARC. And you can see what happens to the microtubules and DNA. In normal cells, the microtubules form a superstructure called a spindle. It's all nicely arranged with two spindle poles at the end. But what happens to the MARC depleted cells is vastly different. There is no structure to it whatsoever. And you can see that's happened to the DNA as well. In normal cells, the DNA is nicely arranged in a straight line, but in the MARC cells, they're just all over the place. But these are just pictures, snapshots in time. What we'd ideally like to do is follow a cell as it goes through cell division. So that's what we did. We took a movie of, we used a microscope to take movies of cells as they go through cell division. What I'm going to show to you here is a normal, unperturbed cell dividing into two daughter cells. I don't know if you can see, but this is how a normal cell looks like before it starts cell division. You can already see the DNA in green and microtubules in red, but they're not very bright. This will change once the cell starts to divide, because the DNA will condense into chromosomes, and the rope-like microtubules will also start to form the structure called a spindle, which will pull the chromosomes apart once they're all nicely attached and arranged at the equator. I'll play that again so you can appreciate it. And notice that the spindle is sort of positioned square in the center of the cell, and it's nicely dome-shaped circle. And the DNA splits quickly and cleanly once it aligns. Here I have a frame-by-frame picture of the video. You can see that the DNA starts to condense at around 4 minutes after the membrane holding the DNA together breaks down. It's quickly aligned at the equator, and within half an hour of the whole thing starting, the DNA has split, and within 1 hour, the daughter cells are staying nicely together in their environment, wherever it may be.